Yes, everyone. So hope you all are here. So a very warm good evening. So hope the video and audio is perfectly clear for you guys. So we are here with yet another new chapter that is biological classification about the module. So right in front of you today we are gonna start biological classification lecture number 1. Yes, yes, Binaya. Yes, Abhijit. So let's start today's session. Yes, Manchudat. So what's your name? I'm Amal. I'm Amal. Okay. So today we are going to start with biological classification lecture number 1. So let's upgrade with Biopoint for the sweet NEET 2023 Biopoint Jwala batch. So are you all ready to start with today's session? So if it's perfectly ready, give me thumbs up on the screen. If you are ready, give me thumbs up on the screen. Yes, Binaya. Yes, come on. Yes, Abhijit. Yes, yes, Abhijit Kumar. Oh, Abhijit Sri Kumar and Abhijit Kumar, two are there. So I will be reading the entire name. Okay. So, biological classification. In this chapter, you will be having all these subtopics. Okay, these 7 subtopics. That is, you will be having the very first subtopic as basics of classification. Second one, kingdom Monera. Third one, kingdom Protista. Fourth one, kingdom Fungi. Fifth one, kingdom Plantae. Sixth one, kingdom Animalia. And seventh one, virus, viroids, and lichen. So these are the 7 different topics that you have, subtopics that you have in this chapter, biological classification. Yes. So in this very first lecture, we are gonna take it together. That is basis of classification and kingdom Monera. In the second lecture, you will be having a single unit or single lecture with Kingdom Protista. The third lecture, you will be having a single module with Kingdom Fungi. In the fourth lecture, you will be having a single session on, in the fourth lecture, you will be having sessions on Kingdom Plantae, Animalia, virus, and virus, viroids, and lichen. Yes, Plantae and Animalia we don't need to learn in detail because we have separate chapters called Plant Kingdom, that is the third chapter, and Animal Kingdom, that is the fourth chapter. So just a brief of Plant Kingdom and Animal Kingdom in this chapter, and after that we will be having the most important topic, virus, viroids, and lichens. Yes, so all the 8 ones, give me thumbs up on the screen. So let's start with the new chapter, biological classification. And right now I'm reminding you that the updated playlist of the 2 chapters, living world and animal kingdom, is given there in the description box. Yes, so do visit that and use it maximum for your further studies if anyone haven't attended the first sessions of any of these chapters. Yes. And now let me remind you to join the Telegram group where you have the daily schedule, where you can interact with me, where you have MCQs in the form of polls, and you get mentor doubt clearance. Yes, it's Sir Aishwari. It's Sir. Yes, Ranjit. Yes, Binaya. Yes, Abhijit Srikumar. So the direct link to join the Telegram group is given there in the description box. That is t.me/neat_biopoint. Yes. This is the topic-wise analysis of the chapter biological classification. Yes, the most important topic, as you can see here, is the virus, viroids, and lichens. As I have told, virus, viroids, and lichens is the most important topic. And after that, this green area refers to kingdom Plantae, kingdom Animalia. Fungi is the next most important topic. The next most important topic in the previous 8 years is kingdom Fungi. And after that is the kingdom Protista. Kingdom Protista. And after that, you have the weightage Kingdom Monera, and at last, you have a least weightage on the topic 5 kingdom classification. This is the 8 years NEET or AIPMT analysis of subtopic wise analysis of biological classification. Yes, everyone. So as per the last 33 years of NEET or AIPMT, you can see that the topic for 36% is virus, viroids, and lichens. Then 19% you are having kingdom Monera. 17% you are having kingdom Protista. 17% in same weightage you are having kingdom Fungi. 2% each for kingdom Plantae and Animalia, and 7% for 5 kingdom classification. As we are analyzing the last 33 years of NEET or APMT, this is the subtopic based analysis of biological classification. Yes, yes, do take the screenshot, or you will be getting at last at the end of entire chapter you will be getting the PDF of the session as you got in the time of living world. Yes, now reminding you to do hit the like button, do share it with your friends, and do subscribe Biopoint, stream the world of bio. So with the full Josh, let's move on with today's session, biological classification. Yes, as we have discussed, the very first topic of this session is basis of classification, and after that we will be moving on with kingdom Monera. So you have to have a clear listening to this session, because some of the points I will be dictating, that is I will be saying right here, and you have to just note it down. Yes, so make a clear lecture note, everyone. So let's move on with today's session, biological classification. The very first is basis of classification. So the basis of classification was done by Aristotle. Yes, Aristotle did the earliest attempt to do classification, was the earliest one to do attempt a more scientific basis of classification. So who did a more, this is the very first question, that is one of the previous year question. Who was the scientist who did an earliest attempt to have a classification on a more scientific basis? That is Aristotle. That is, he divided plants into trees, shrubs, and herbs. As you all know, trees, shrubs, and herbs are the very basic classification of plants, and that was done by, that was the earliest classification on a scientific basis done by Aristotle. And he classified animals into two, that is animals that are having red blood and animals that are do not having red blood. Okay, so animals that are having red blood, he gave a name, anemia. Anemia. And animals that do not have a red blood, they gave the term, he gave the term anemia. Yes. So as you all know, in the same similar conditions, we are saying that if a person is deficient of or deficient of hemoglobin in their blood, we will be saying that person is pale, that is he is anemic. So without red blood, that is red blood pigmentation is by the what hemoglobin. So due to the pale, just connecting with that. Okay, if there is no red blood in animals, he gave the term anemia, and if there is red blood, he gave the term anemia. Yes, yes. Be active in the comment box. I am not receiving any chats. Yes. Okay. So that was the basis of classification. And after that, the next system of classification is done by Linnaeus. The next system of classification is done by Linnaeus. That is a modern basis of classification, two kingdom system of classification. So before moving on to that, let me give a brief description to you about this classification. Yes. So if we are saying about biological classification, I'm just denoting it by the term BC. Okay, because if I am going to write it in detail, my handwriting will be getting more worse and worse, because the pen is not at all working properly right now. Yes. So biological classification was done, was classified, first classified into two. That is, we are just giving an outline of all the classification. Who is Abhi sir? It's another sir who is taking, who is gonna have you class. Who is gonna take you class right at 9 p.m. the next new chapter, digestion and absorption. Yes, yeah, I can understand that, Sankita. So, the basis of classification is as ancient and modern. This is a basic outline. Modern classification. Okay, you may leave. Okay, so the ancient system of classification is given by two scientists, that is Aristotle. Guys, are you getting this? No one is commenting. Aristotle and Theophrastus. Aristotle and Theophrastus gave this, yes, Abhijit Sri Kumar, gave this ancient system of classification. Yes, Abhijit Kumar. Binaya, Lucifer, Ranjit. Oh, all of you are here. Yes, be active. So, modern system of classification, if you are going to see in detail, it can be further classified into three. Modern system of classification can be further classified into three, that is as artificial system of classification, natural system of classification, and phylogenetic system or phylogenic system of classification. Phylogenic system of classification. Yes. So artificial system of classification is proposed by Linnaeus. Natural system of classification is proposed by Bentham and Hooker. Two scientists, Bentham and Hooker. Yes, everyone. Phylogenic system of classification is proposed by four scientists, that is Enkler and Prantl. Enkler and Prantl. Yes, I'm not getting space there, so I'm going to write here. Okay. Second, the third one is Hutchinson. By heart, the name of all these scientists, all these are important. Hutchinson. Then the last one is Takhtajan. If anyone needs this spelling, please comment it. I will just post it in the chat section. Takhtajan. Yes. So ancient system of classification is proposed by two scientists, that is Aristotle and Theophrastus. Modern system of classification is done by, modern system can be further divided into three, that is artificial, natural, and phylogenic. Yes, artificial system of classification is done by Linnaeus. Natural system of classification is done by Bentham and Hooker. Phylogenic system of classification is done by Enkler and Prantl, Hutchinson and Takhtajan. Is it clear? So hope I can change the slide. Yes, because we are having Menti right at the end, so I have to complete it so too faster. Right now it's 7:17. Yes, fine. So two kingdom system of classification that is proposed by Linnaeus. So the two kingdom system of classification comprise of two kingdoms, that is Plantae and Animalia. Kingdom Plantae and Kingdom Animalia. That is what about two kingdom system of classification. Yes. Is it clear? Then we are moving on with the next, that is drawbacks of this two kingdom system of classification. So if we are saying about the drawback of two kingdom system of classification, that is prokaryotes and eukaryotes were included in a same kingdom, that is under plants. Both prokaryotes and eukaryotes were included in the same kingdom, that is under the kingdom Plantae, and because they showed the presence of cell wall. That is why they were included in the kingdom Plantae. Yes, but they had a vast difference in many other features. But they were included in kingdom Plantae, so that was one of the main drawback. Second one is that both unicellular and multicellular organisms were placed in the same group. Unicellular, multicellular organisms were mixed and placed together in a same group, that is under algae. Okay. So, and this two kingdom system of classification does not differentiate between heterotrophic fungi and autotrophic green plants. They included all these plants together. Both heterotrophic fungi and autotrophic green plants, all these were included together in this two kingdom system of classification. Okay. Fungi have a chitinous cell wall that we will learn in the upcoming slides. I mean, upcoming sessions. Kingdom fungi. Session of kingdom fungi, we will learn that. And green plants have a cellulosic cell wall, but all these were included together. Therefore, we cannot say this two, we cannot accept this two kingdom classification as a permanent one. So after that, further classification came. Before moving on to this five kingdom classification, there were further classification came. That is three kingdom classification. Three kingdom classification proposed by Ernest Haeckel. Ernest Haeckel. Oh my god. Ernest Haeckel. Yes, Ernest Haeckel proposes three kingdom system of classification. So other than Plantae, Animalia, kingdom Plantae and kingdom Animalia, the new one that was added was kingdom Protista. Kingdom Protista was added. Yes, kingdom Protista was added in this three kingdom system of classification. So after that came the four kingdom system of classification. Four kingdom system of classification was given by Copeland, a scientist called Copeland. Yes, four kingdom system of classification was given by a scientist called Copeland, and the new kingdom that was added was kingdom Monera. Kingdom Monera was added in this four kingdom system of classification. Okay, so after this four kingdom came the five kingdom system of classification to differentiate kingdom fungi that was given by a scientist called R.H. Whitaker. So we have to learn in detail. In this entire chapter, we are going to learn in detail about this R.H. Whitaker and its five kingdom system of classification. Yes. Which one, Abhijit? 1894, which one? So, R.H. Whitaker proposes five kingdom classification in 1969. Very important year. 1969, R.H. Whitaker proposed this five kingdom system of classification. So before moving on to detail of this five kingdom system of classification, another system of classification came. That is the sixth kingdom that we are not going to learn. The mostly accepted one is five kingdom system of classification, and sixth kingdom also rose up, and that was given by a scientist called Carl Woese. Carl Woese gave the sixth kingdom system of classification. Yes. So in this sixth kingdom system of classification, he just removed kingdom Monera. That is, he just removed kingdom Monera by splitting it into two. Yes, three kingdom was given in the year 1866. Abhijit, 1866. It was not 1894. The year that you have to learn is only 1969. That is the year for five kingdom system of classification. Yes. So Carl Woese gave the six kingdom system of classification. In that, he splitted kingdom Monera into two. That is as Archaebacteria and Eubacteria. Archaebacteria and Eubacteria we will learn today itself. Okay, Archaebacteria and Eubacteria. Okay, etc. Okay, so this was the further systems of kingdoms of classification. Yes, so let's move on with today, that is the five kingdom system of classification. Yes, everyone. So, five kingdom system of classification was proposed by R.H. Whittaker in 1969. It includes five different kingdoms, that is Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. Yes, so if anyone want to remember these five kingdom systems of classification, use the clue map of places. Map of places. What all are the kingdoms coming under this five kingdom system of classification? Map of places. Oh my god. Okay. So map of places. M refers to kingdom Monera. P refers to kingdom Protista. F refers to kingdom Fungi. P refers to kingdom Plantae, and A refers to kingdom Animalia. This is what is what said as map of places. That is five kingdoms of classification: Monera, Protista, Fungi, Plantae, and Animalia. Yes, everyone. So this classification was based on, very important, what were the criterias? Yes, question for both boards and NEET examination. What are the criterias that Whittaker used for his five kingdom system of classification? Yes, so Whittaker used certain criterias like cell structure, thallus organization, mode of nutrition, mode of reproduction, and phylogenetic relationship. These were the different criteria that Whittaker used for his different systems of classification. That is the cell structure of their cell. Yes, whether that is their compositions, their outer layers, their inner layers, all these were considered under the cell structure. Then thallus organization. Thallus organization, that is their body is differentiated into head, thorax, and abdomen, like that different what differentiation of their bodies. That is what is said as thallus organization. Mode of nutrition, whether it is autotrophic, heterotrophic, chemosynthetic, everyone. Yes. Reproduction, asexual or sexual or vegetative propagation. Phylogenetic relationship, that is evolutionary relationship, that is due to a heredity, ancient relationships. All these were considered under the classification of five kingdom system. Yes, everyone. Only Binay is commenting. Lucifer, Rajni, Abhijit Sri Kumar, Abhijit Kumar, everyone. Shravani, yes. So these were the criteria that Whittaker used for his five kingdom system of classification. So let's analyze that in detail. So cell type, that is, or the cell structure. Yes. Cell type, that is kingdom Monera, is prokaryotic. Phylogenetic refers to evolutionary, Binaya, evolutionary relationship. Okay, evolutionary relationship is what is meant by phylogenetic, that is due to their ancient heredity. Transmission of characters, that is what you have learned in heredity and evolution class 10, right? So like that, the ancient transmission of characters from which, what was their origin, that what was their relation with the previous organisms. Yes. So only kingdom that is prokaryotic, comprising of prokaryotic organisms is Monera. Prokaryotic refers to unicellular, right? Unicellular organisms is what is meant by prokaryotic. So Protista, fungi, Plantae, and Animalia, all these are comprising of eukaryotic organisms. Yes. Cell wall. This next, if you are more going to say about cell wall in case of Monera is non-cellulosic. All those composition we will learn in detail. All these topic we will learn in detail in different subkingdoms of Monera, Protista, Fungi, Plantae, and Animalia. Yes, everyone. So right now let's move on with kingdom Monera. Everyone, ready for that? Let's move on with kingdom Monera. So if you have understood everything clearly till now, give me hearts on the screen. Give me hearts, guys. Give me hearts, guys, fast. So let's move on with Kingdom Monera. Bacteria, that is the unicellular organism. So the most abundant microorganism in this under this kingdom Monera is bacteria. Yes. So hundreds of bacteria are present in a handful of soil. If we just take a handful of soil, we are just getting hundreds of bacteria, right? So they can survive in hot springs, deserts, snow, and deep oceans. And they are many of the bacteria are parasite. That is, they obtain their food from a host body, from others' body. Yes, that is all about the basis of kingdom Monera. So if you have doubt with any single term, you should ask, because this chapter forms the basis for many other chapters. Okay, plant kingdom, animal kingdom, everything is based upon this chapter. Microbes in human welfare, human health and diseases, that is the diseases caused by these bacteria. All this you should have know about all these bases. So everyone, if you are having any doubt with any of the topic, you just comment me right at the point when you're arising with the doubt. Yes. So moving on with kingdom Monera, comprising of unicellular organisms or the only prokaryotic kingdom. So as you all know, we have learned all this in what previous lectures. So all members means only member. The only member of kingdom Monera, that is bacteria. Why are they called most abundant? Because the most varieties, most amount, most quantities of bacteria can be seen. For example, as we have said, if you are just taking a handful of soil and if you are just analyzing it under a microscope, you can see a vast varieties of bacteria in that. Yes, therefore it is most abundant. They divide faster due to their division of binary fission, right? Therefore, they are the most abundant. They are said to be the most abundant, and the sole members means that it is the only member of Kingdom Monera. Clear, Abhijit and Shravani? Yes. So, coccus, that is a spherical bacteria, rod-shaped bacillus, comma-shaped vibrio, and spiral-shaped spirillum. Abhijit, Shravani, it's okay for Shravani, then Abhijit. So this is all about the four types of bacteria. Spherical coccus, rod-shaped bacillus, comma-shaped vibrio, and spiral-shaped spirillum. Yes, Abhijit. Okay, fine. So now we are more going to move in detail with the analyzing different structures, their body organization, then their mode of nutrition, mode of reproduction, all these things that we are going to learn in the upcoming slides. Yes. So bacterial structure is very simple, but they are very complex in behavior and show metabolic diversity. Yes, some bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. Majority are heterotrophic, so some are autotrophic, that is, they synthesize their own food, that is, some are chemosynthetic autotrophs, true, that is, they using certain chemicals they prepare their own food. Yes, so some of them are autotrophic and some of majority are heterotrophic, that is, they obtain their food from a host body. Yes. So have you all got the NCRT of this chapter? Have you all got the NCRT of this chapter? PDF of NCRT? Guys, come on. So further classification of bacteria as to archaebacteria and eubacteria. So wait one second. Let me check whether this is all about bacteria. Before moving on to this types of bacteria, let me give some more details about this basis of bacteria. Okay. No, sir, I have book. Okay, Abhijit. Yes, I understood because you asked just all members. That is a statement of NCRT. I just thought we were having this NCRT. After this session, we will be posting this NCRT. Okay, guys. So these are unicellular, right? Kingdom Monera is unicellular, and their cell wall is made up of, we said that their cell wall is non-cellulosic, that is, it is not made up of cellulose. Then what is the composition of their cell wall? Their cell wall is made up of peptidoglycan. Their cell wall is made up of peptidoglycan. Yes, and this peptidoglycan 2 is having two exceptions. That is, in case of archaebacteria, that we will learn what is this archaebacteria. That is a classification. Okay, archaebacteria, in case of archaebacteria, and in case of mycoplasma, these two things we will cover today itself. Okay. Yes, Ishika. Archaebacteria and mycoplasma, this cell wall is made up of other constituents, that is other constituents, and in case of this kingdom Monera, all other members of kingdom Monera, it is made up of peptidoglycan. And this peptidoglycan is composed of two further constituents. Very important, previous year need questions. NAM and NAG. NAM and NAG. These are the two further constituents, two further compositions of peptidoglycan. Is it screen clear, guys? Is the screen clear? NAM and NAC. So the full form of this NAM is, what do you mean by NAM? It is N-acetyl muramic acid. The statement will be given in this chat section. Okay, if I am writing it fully, the screen will be just becoming overflowed. Okay, N-acetyl muramic acid. That is the full form of NAM. And in case of NAG, it is N-acetyl glucosamine. N-acetyl glucosamine and N-acetyl muramic acid. Then the next point is that all membrane-bound cell organelles are absent. That is, there is no membrane-bound cell organelles. No membrane-bound cell organelles are present. Yes, instead, 70S ribosomes are present. That is a type of ribosome. 70S ribosomes are present in case of this kingdom Monera. 70S ribosomes are present. Ribosomes are non-membranous, right? So, non-membranous organelle, that is 70S ribosome, is present in case of kingdom Monera. Yes, and the reproduction of this kingdom Monera is asexual. This is all about the extra points that I'm planned to give to you. All about these extra points. That is, they are unicellular. Their cell wall is made up of peptidoglycan, which is composed of NAM and NAG, N-acetyl muramic acid, and N-acetyl glucosamine. And this peptidoglycan is having an exception, that is in case of archaebacteria and mycoplasma. And we have said that no, there is no membrane-bound organelles than their presence of 70S ribosomes. Yes. So now we are moving on to the classification of kingdom Monera. That is, bacteria can be classified into two: archaebacteria and eubacteria. Archaebacteria and eubacteria are the two classifications of this kingdom Monera. Yes, archaebacteria, they are having an incipient nucleus. They are known as the pseudobacteria or a false bacteria. Okay, pseudobacteria, that is a false bacteria. Archaebacteria is known as a pseudobacteria because their cell wall composition is different, right? And this eubacteria is known as true bacteria. True bacteria, pseudobacteria and true bacteria. Everyone, is it clear? Guys? Yes. So let's move in detail. On that, we will learn about this further classifications. Archaebacteria is further classified into three: halophiles, thermoacidophiles, and methanogens. Eubacteria as autotrophic bacteria and heterotrophic bacteria. Autotrophic can be further classified into two: photosynthetic autotroph and chemosynthetic autotroph. And heterotrophs can be classified into two, that is parasitic heterotrophs and saprophytic heterotrophs. Yes, Binaya, Abhijit, and Shravan. Yes. So archibacteria, they can be archibacteria, as we have discussed, they can be classified into three: halophiles, thermoacidophiles, and methanogens. So one important point on this about this case of archibacteria is that they are the oldest living fossils. Very important previous year NEET question. Oldest living fossil is archibacteria. They have been found from a few most of the years back. Okay, some huge period of time since some huge period of time this archibacteria is present. So therefore, it is known as oldest living fossil. And they lack peptidoglycan in their cell wall. Instead, their cell wall is made up of branched lipids. Instead, the cell wall of archibacteria is made up of branched lipids. Yes, each and every point I am including in this chapter, biological classification, in the same way as I have taken living world. So, if you are just making lecture notes by this, you will be having a perfect score for your boards and for your NEET. Yes, because there is no other points to be asked out of this topics. If there is any point extra from the slides, I'm just giving to you. Yes. So this can be further classified into halophiles, thermoacidophiles, and methanogens. Halophiles as extreme salty areas. Bacteria that are found in extreme salty areas is known as halophiles. Hot spring, that is bacteria that are found in hot springs, it is known as thermoacidophiles. Bacteria that is found in marshy areas is known as methanogens. Everyone? Yes. So, archibacteria have a different cell wall structure for their survival in extreme conditions. And first one is methanogens, that is, they are found, very important point, they are found in the gut of ruminant animals, such as cows, buffaloes, in their gut. Look here. In their gut, this bacteria called methanogens are found. So, cell wall is made up of branched lipid. Yes, I will give it in the chat box. Branched lipids. Yes, Vinaya. Okay, fine. So these methanogens are found in the gut of ruminant animals and they produce the methane gas, that is a biogas from the dung of animals. So we can see that we always have heard that from the cow dung we are obtaining methane gas. Yes. So this gut of ruminant animals, they contain the bacteria called methanogens and these help in the production of a biogas called methane. So next is the eubacteria. That's all about archibacteria. Yes. Next is the eubacteria. Eubacteria is the true bacteria having a rigid cell wall and a flagellum. That is, if motile, they have a flagellum. If it is non-motile, it lacks flagellum. Yes. Okay. They include autotrophs, that is photosynthetic and chemosynthetic autotrophs and heterotrophs. This is about the classification of eubacteria. Is this chapter easy? Are you able to follow up with me? Are you able to follow up with me? Yes. Okay, fine. So, bacteria can be classified, eubacteria can be classified as autotrophs, that are photosynthetic autotrophs, very first classification. Yes. So, they are having chlorophyll A that is similar to that of green plants. So, the chlorophyll that is present in green plants is chlorophyll A. That same chlorophyll is present in photosynthetic autotroph, that is in the case of cyanobacteria. Cyanobacteria is an example for photosynthetic autotroph. Okay, cyanobacteria is a what example for a bacteria, that is a photosynthetic bacteria consisting of chlorophyll A, that is the chlorophyll similar to that of the leaves of green plants. Cyanobacteria is also known as blue-green algae. Yes, and it is unicellular, colonial, or filamentous. They can be seen as a filament like this. They are seen in colonies, that is, they are seen together. They are seen in clusters, they're like in colonies. Okay, they are unicellular, they are single celled. Yes, they are marine or terrestrial. They can be seen in water, they can be seen in land. Okay, therefore, they are known as cyanobacteria or the blue-green algae, and their colonies are generally surrounded by a gelatinous sheath. Their colonies are generally surrounded by a gelatinous sheath. Yes, and they often form blooms in polluted water bodies. Most of the polluted water bodies we can see the cyanobacteria. Yes, and their major agricultural importance is they help in nitrogen fixation because they contain certain cysts in them in their body, specialized cell in their body. There are specialized cells in their body called heterocysts that help in nitrogen fixation. Yes, example is nostoc and anabena. Nostoc and anabena is the example for cyanobacteria. How does gelatinous sheath look like? It's just an outer covering. As usual, we are having a covering, right? Like that. It is an outer covering. That is what is said to be gelatinous sheath. That is how we can say a gelatinous sheath looks like. An outer covering. Yes. In this outer covering, they will be comprising of all the colonies, not a single cyanobacteria or colonies will be there. Okay, enclosing all the colonies. For example, if you are getting, if you are observing a pen, we are buying a packet of pens. All those pens, individual pens will be individual cyanobacteria, and those pens put in that cover, right? That cover is shown as the gelatinous sheath. Okay, this is the very basic explanation. Yes. Okay, fine. So an example, what for blue green algae or cyanobacteria is nostoc and anabena, and what I have to say is the first oxygen evolving organism. The first organism from which oxygen evolved is cyanobacteria. First oxygen evolving organism. Clear, guys? Which was the first oxygen evolving organism? It is cyanobacteria. Yes. So we are moving on to the next topic. Oh my god, chat is getting stuck. Okay, fine. Okay, next is this chemosynthetic autotroph. They oxidize inorganic substances such as nitrate, nitrite, and ammonia, all these different chemical substances to release ATP energy for ATP production. That is, they release their energy by breakdown of nitrate, nitrite, and ammonia, and they help in recycling of nutrients like nitrogen, phosphorus, iron, and sulfur. All these are just recycled by half used nitrogen. Is just half used nitrogen is just recycled by what this chemosynthetic autotrophic bacteria. For example, green sulfur bacteria is an example for chemosynthetic autotrophic bacteria. Purple sulfur bacteria. These are the two basic examples for chemosynthetic autotrophic bacteria. Is it clear, guys? Is it clear, everyone? We are having four more slides, and after that, we will be having the Mindy Quest. Everyone must join. Now I am receiving only in the upcoming what chat section. I am receiving only chats from Abhijit Kumar, Binaya, and sometimes Shravani. And where are the others? Lucifer, Rajneet, everyone. Be an active in the comment section. Okay. Yes. So next is the heterotrophic bacteria. Guys, are you all understanding? By seeing the chat box remaining silent, I'm just what thinking that you are not getting the points that I'm saying. Yes. Is it like that? Or is the audio stick? Or is the audio stick? No one is commenting. What can I do? Yes. Okay. Fine. 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 Now only the chat got refreshed. Yes. Chemosynthetic autotroph once more. Okay, fine. Okay, thank you, thank you, thank you. All of you should stay here until the Mindy quiz. All of you should attend the Mindy quiz and should know your position in that. Yes? Okay. So chemosynthetic autotroph means that certain chemical substances like nitrate, nitrite, and ammonia. Okay. So bacteria just oxidize. They just break down these nitrate, nitrites, and ammonia for releasing energy. That is for ATP production. Yes, that is for their energy production. And examples for chemosynthetic bacteria is green sulfur bacteria, purple sulfur bacteria, and certain chemical component elements like nitrogen, phosphorus, iron, and sulfur. They also can be recycled by chemosynthetic autotrophic bacteria. Yes, Vinaya. Next is the heterotrophic bacteria. That is, they are the most abundant in nature. We said that bacteria is most abundant in nature. From that, majority of the bacteria is heterotrophic. That is, they obtain their food from a host body. Yes. Okay, fine. So more majority are important decomposers. As all of you know, bacteria decompose. So what? That is dead and decaying organism to convert it to an organic matter. All these are done by heterotrophic bacteria. They too are useful. There are both useful and what non-useful bacteria there, right? In every case, there will be an equal and opposite what types of organism. Okay, so impact of heterotrophic bacteria in human affairs. That is, they help in the conversion of curd from milk. Conversion of curd from milk. So this conversion of curd from milk is done by a bacteria called lactobacillus. Lactobacillus bacteria help in the conversion of curd. I mean curd from milk. Yes, exactly. Yes, and production of antibiotics. Different bacteria, their inactive form can be used in the production of antibiotics. Fixation of nitrogen in legumes, legume roots. Yes, fixation of nitrogen in legume roots such as rhizobium. Rhizobium is a bacteria that help in the fixation of nitrogen in legume roots. Yes, and some pathogens, that is the harmful effect that they cause diseases like cholera, typhoid, tetanus. All these are in animals and citrus canker in plants. These are different varieties of diseases caused by this bacteria. Yes, so there are both useful and harmful effects of this bacteria. Yes, everyone. Reproduction of bacteria. Bacteria mainly reproduce by fission. Under unfavorable condition, they produce spores. And they also reproduce by a sort of sexual reproduction, that is DNA transfer from one bacterium to another. Okay, just by transferring. Look here, DNA is just going to getting transplanted. Yes. So, different types of reproduction in this bacteria is the bacteria mainly reproduce by fission, that is the asexual mode of reproduction. In rare cases, that is in under unfavorable conditions, they produce spores, and in some cases, they reproduce by a sort of sexual reproduction, that is just by DNA transfer from one bacterium to another. Yes. Next is the last and final topic, mycoplasma. Mycoplasma. Mycoplasma, also known as PPLO. PPLO. Mycoplasma is also known as PPLO. Yes, pleuropneumonia like organisms. That is the full form of PPLO. Pleuropneumonia like organisms. Pleuropneumonia like organisms. It will be just posted in the chat section. That is what is meant by PPLO. So mycoplasma is also known as pleuropneumonia like organisms and they are organisms without a cell wall. So these organisms are without a cell wall and they are the smallest living cells ever known. Till now, the smallest living cells that are known as Mycoplasma. And this Mycoplasma is discovered by two scientists called Knockard and Rocks. Not important, but anyway, just keep it in your mind. If it is asked for your entrance examination, it will be useful. Knockard and Rocks. Okay, and these mycoplasma can survive without oxygen. Many are pathogenic in animals and some are in plants. Okay, so this mycoplasma lack their cell wall. They are the smallest cell ever known, and their colony looks like a fried egg manner. Okay, and what else to say? They were discovered by Knockard and Rocks in 1898. Year is not at all important, that's why I'm not mentioning. Yes, and who gave the term mycoplasma? The term mycoplasma was given by a scientist called Novak. Novak, Nowak, Novak. He gave the term mycoplasma. Okay, here I am not mentioning because if I am mentioning the year, it will be getting confused for you. It is not important. Okay, here is not important. Yes, and this mycoplasma is known as jokers of the plant kingdom. Mycoplasma is known as jokers of the plant kingdom. Yes, jokers of the plant kingdom. That is Mycoplasma. And that's all. Now we are moving on with Mindy Quest. Everyone, stay tuned with us. Let's move on with the Mindy Quest. Yes, go to www.minty.com and use the code 16265719. Yes, 16265719 is the Minty code. Yes, 16265719 is the Minty code. Join fast. So we are moving on with today's Minty Quest. Yes, join guys using the code using the link www.minty.com and the code 162657. Keep joining. Join fast. Come on. Time is going. So if you join fast, we can move fast to the further questions. Yes, come on. I'm not receiving your chats. I hope you all have entered this. Only five here. Eight were there in the session. Yes. So we are gonna start today's Minty quiz. Yes. Answer fast to get more points. Two kingdom system of classification was first proposed by Aristotle, Carl Linnaeus, Bentham and Hooker, R.H. Whittaker, who proposed the two kingdom system of classification. Yes, everyone has voted. The voting rate. One has made it wrong as Aristotle. I don't know who is that. Five made it right. Well done. Yes. The second question. Answer fast, you get more points. Five kingdom system of classification was given by R.H. Whittaker in the year 1822, 1869, 1929, 1969. Yes, everyone voted it right. Well done. Yes. So let's move on with the third question. Answer fast to get more points. Cyanobacteria is used in agricultural fields for crop improvement because they cause nitrogen fixation, algal blooms, photosynthesis, all of these. We have just discussed it due to the what presence of special spores, what cells to made it wrong. All the options are wrong. Oh my god. It was the issue with Mentimeter. Very sorry for that. So let's move on with the next question. The fourth question on the screen. Yes, answer fast to get more points. Methanogens can be observed in. Yes, hot springs, extreme saline habitats, gut of ruminants, freshwater resources. Yes, methanogens can be observed in. Everyone has watched it. Let's check. Yes, everyone made it right. Well done. Last and final question. Last and final question of the day. Let's check it. Answer fast to get more points. Prokaryotic smallest cell without a cell wall. That's the last topic that we have completed today. Blue-green algae, cyanobacteria, lichens, mycoplasma. Blue-green algae, cyanobacteria, lichens, mycoplasma. Yes, everyone has voted. Yes, again one made it wrong. All the options are wrong. My god, it's the issue with Mentimeter. Yes. Come on. Yes, Amitav Biopoint student is the fastest. Akhil is the second. Shravani Sardi is the third. PV Kuntu is fourth. Sebastian is fifth. And Abhijit is sixth. Yes, everyone, congratulations for your participation. Thank you, guys. Thank you all. Thanks for your patient listening. Do hit the like button, do share with your friends, and do subscribe Biopoints in the world of bio. And at right at 9 p.m., you will be having a live session on biological classification. There were two questions wrong. That is with the Menti problem is with the Mentimeter. Yes. So hope you all have enjoyed today's session. And that's all for today. It's me signing out from your favorite and my favorite Biopoint. Stream the world of bio. Bye bye.